right, at this point, I'm going to introduce our next first-generation leader speaker from our first-year class, which is Andrew Miri. Andrew received his undergraduate degree in neuroscience and mathematics from Brown. He then spent some time as a high school mathematics teacher in Boston, after which he got his PhD from Princeton University, where he used calcium imaging and cell-attached recordings in behaving larval zebrafish to address the circuit architecture of the oculomotor neural integrator. Since completing his dissertation, he did a postdoc at Columbia, and he's just about to start opening his own lab at the neurobiology department at Northwestern University. So please join me in welcoming Andrew.
Thanks for the kind introduction. And I wanted to start by saying, let me get to there. That's a nice one. Let me start by just saying a big thanks to everybody here at Allen. It seems like it's going to be super fun being a part of the NGL Council, and I'm very grateful for the chance to present some of my work to you today. So my general research interest is in the neural mechanisms underlying the execution of movements, like the examples pictured here. And I chose each one of these pictures because I think each one really well captures the really impressive complexity and agility of mammalian movements. And we know that this complexity and agility are both really critical to the fitness these movements confer, and so to the evolutionary success of many mammalian species. And in thinking about the neural mechanisms underlying these sorts of movements, the influence of the cortex, which is elaborated extensively over mammalian lineages, seems really critical. For example, the seminal lesion studies of Lawrence and Kuipers in the 1960s first implicated the corticospinal pathway in particular in enabling both the complexity and agility of, in that case, primate movements. That said, it's true that today we lack a very clear view of when and how motor areas of the neocortex and other subcortical motor areas influence the activation of muscles in real time as movements like these are executed. So my general approach is to take advantage of some of the tools that can be readily brought to bear in the mouse nowadays to try and refine our models of motor system function. So here's a cheetah in the midst of executing movement. And here we can distinguish two phases of the movement that should differ in terms of their requirement for involvement by the motor cortex. At first, the cheetah here is performing a fairly stereotyped locomotor behavior, but then this visually guided component emerges as the cheetah tries to catch this ball that it's chasing. The stereotyped locomotor behavior, that shouldn't require motor cortex. We know that a cat lacking a motor cortex can still locomote. But behaviors like this, the visually guided attempt to catch a ball like that, other experiments suggest that these sorts of movements do in fact require a motor cortex. Now, sort of despite this sort of functional distinction here, we don't actually know what's different, if anything really, about what the motor cortex is doing in real time as phases of movement like this are executed. So let me explain a bit more here. So previous lesion and inactivation studies have led to this view that the role of the motor cortex in driving movements may be fairly specialized, limited maybe just to the generation of movements that require novel combinations of muscle activation patterns or some sort of sensory-guided modification. This monkey is missing the left half of his motor cortex, yet he's still able to climb, also walk around, jump around, and so forth. Might do so with somewhat less agility, but he's still able to generate these basic motor behaviors. Oops, that's too far. This rat here, similarly, has learned to perform a precisely stereotyped forelimb movement in order to get a reward. And when this rat first returns to its behavior box, after having its entire motor cortex removed bilaterally, the rat still performs that same stereotyped forelimb movement, really almost identically to the way it did before the lesion. However, electrical recording and electrical stimulation results suggest that the motor cortex could be playing a much broader role. So electrical recordings show that motor cortical neurons are clicking away in patterns that correlate with muscle activity during a broad range of behaviors, including behaviors like just walking on a treadmill that survive motor cortical inactivation. We can see these sorts of correlations in these activity traces here collected from a limb muscle and a corticospinal neuron as a cat just walks along a treadmill. It's also true that perithreshold electrical stimulation in motor cortex during movements like this causes muscle contraction at short latency. So here I'm showing you a limb muscle EMG recording, again, as a cat just walks along a treadmill. And this bit of stimulation here in motor cortex leads to an extra twitch in the muscle down here. So it doesn't seem like cortical output is somehow just gated out from downstream circuits during behaviors of this sort. So, how can we reconcile these results if motor cortical neurons are always clicking away in patterns that resemble muscle activity, and their output seems capable of driving muscle contraction? How can the same neurons, synapsing on the same downstream targets, make very different functional contributions during different types of movement? Ultimately, we'd like to have a theory of motor cortical function that can account for the actual spikes that motor cortex fires. So today I'll tell you a little bit about how we've taken advantage of some of the opportunities we have in the mouse to begin to clarify a bit about the when and the how of motor cortical influence. So we wanted to take a comparative approach. So we first identified two behavioral paradigms that differ in terms of their requirement for motor cortical involvement. We developed a paradigm in which mice are trained to perform a precision forelimb movement. That's what I'm showing you here in this video on the left. In this paradigm, mice are trained to hold their paw on a rung, and then when a joystick is rapidly positioned, to reach out and grab it and pull it a set but short distance in order to get a reward. If the pull is a little bit too much shorter or too much longer than the goal, which in this case is about 5 millimeters, the mouse doesn't get a reward on that trial. And I'll point out that this behavior here was designed to mimic those that are known to require motor cortex, even in rodents. Rodents can still move their limbs and push levers in stereotyped ways without a functioning motor cortex. But there's a lot of data now from labs like Ian Wishaw's and Ben Sylvetsky's suggesting, that more precision forelimb movements like this do in fact require motor cortex, even in rodents. And we've been able to show, in our case, that this behavior does in fact require motor cortex using both the unilateral injection of the GABA agonist mucimol and the unilateral ablation of motor cortical tissue. Here I'm plotting the behavioral performance of a representative mouse across behavioral sessions. And the performance in this task is drastically diminished by the injection of mucimol, but not by the injection of saline alone. And unilateral ablation has a similar effect on behavior. OK, so now getting mice to do something which should not require motor cortex, that's much easier. We can just put the mouse on a treadmill and turn it on. That looks like this. And mice still walk fine on the treadmill even after inactivation of motor cortex. You can see this here in these average EMG traces recorded from 6 forelimb muscles during treadmill walking in a mouse, both before and after the injection of mucimol. The behavior really looks essentially identical. And here again, we saw similar results with ablation. OK, so how do we then account for the specificity of inactivation deficits seen by us and many others during tasks like this? Well, one basic possibility is that motor cortex drives muscles similarly across a broad range of behaviors, maybe all behaviors, but it's the rest of the motor system that's only able to compensate for the loss of motor cortex during a subset of movements, because its functionality is somehow limited. It's also possible, though, that the specificity of inactivation deficits reflects a selective influence of motor cortex on muscle activation during particular types of movement. So we addressed this using transgenic mice that express chanorodopsin 2 in most all of the inhibitory interneurons in cortex. This is the VGAT chanorodopsin 2 transgenic line. Then we can shine a spotted blue light onto the surface of the brain, covering most of the forelimb area of the primary motor cortex, what's called the caudal forelimb area in mice. In doing so, we'll very quickly and almost completely turn off the output from that chunk of tissue. Here I'm showing you results from some silicon probe recordings that were done in the deep output layers of motor cortex during chanorodopsin stimulation here. We find putative GABAergic interneurons that are activated by each blue light pulse. But the majority of neurons, including putative pyramidal neurons, show a rapid and nearly complete cessation in their firing. OK, so what happens when we do this while the animals are performing that precision pull task? Well, what I'm plotting here in gray is the trial averaged EMG for 2 different limb muscles in one of our mice as it performs the precision pull task. In blue, we have the same kinds of EMG trial averages, but for trials when the blue light stimulation was applied at 20 hertz. In the center here, use trials when the blue light comes on just after the animal starts to reach towards the joystick. And on the right here, the blue light came on a bit later during the behavior, just after the animal starts to pull the joystick. In both cases here, the effect of an activation is extremely fast. It takes about 10 milliseconds for the EMG measurements to diverge from controls after the blue light comes on. Okay, so what happens during treadmill walking? Well, here we see something different. The motor behavior is not immediately perturbed when the blue light comes on. Here I'm showing you data now from 3 different muscles in one of our mice. And you can see that the EMG trial averages with and without blue light stimulation, they don't immediately diverge when the blue light comes on, although often there is some divergence a bit later during the behavior. And we can quantify these effects over time. So here I'm showing you the difference between control and inactivation trials summed across all 6 of the forelimb muscles that we recorded from in our mice. During walking, instead of an effect setting in at just about 10 milliseconds, we don't see much of an effect until about 35 milliseconds here. And this effect at 35 milliseconds is actually not prominent at all phases of the step cycle during treadmill walking. When we do inactivations instead at all phases of the step cycle and then average all the results together, we see an effect that's comparatively small here. But OK, so what does this difference in latencies actually mean here? Well, the effect at 10 milliseconds here appears to indicate that relatively direct pathways leading to muscles are engaged by motor cortical output selectively during that trained precision pull behavior. The evidence for that comes from electrical stimulation experiments that we did. So it turns out that when you electrically stimulate in motor cortex in a mouse, the first responses that you see in EMG recordings from limb muscles are just about exactly 10 milliseconds later. Here I'm showing you average change in muscle activation, again, summed across all of the muscles that we recorded from during electrical stimulation in one of our mice, and you can see the effect setting in here at just about 10 milliseconds. And we think that the earliest responses there at 10 millisecond reflect stimulation of the shortest latency pathways, or the shortest pathways connecting the motor cortex and spinal motor neurons. So that's going to include corticospinal neurons that synapse onto spinal interneurons, that in turn project to spinal motor neurons. There's also cortical output targeting the brainstem, where there are neurons that project directly to spinal motor neurons as well. And thinking back to our cheetah here, we might imagine now that at that transition from stereotyped locomotion to the visually guided attempt to catch the ball, that these short latency pathways are suddenly getting activated. The question then is, well, how? You know, as I said, previous observations would suggest that motor cortical neurons should be active in patterns that correlate with muscle activity, seemingly during any kind of movement. Okay, but there must be something about the structure of that activity that allows this behaviorally selective pathway engagement. So we wanted to try and figure out what that structure and activity could be. So we wanted to look at activity in the same motor cortical neurons during both the precision pull behavior and during treadmill walking. We used tetrodes to record from neurons in the deep output layers of mice. And across several behavioral sessions, we were able to aggregate recordings from several hundred individual neurons in each of our mice. Did a lot of analysis of this data. The analysis argued against a number of hypotheses we considered for differential influence that I'm going to have to skip for the sake of time today. But the mechanism that seemed most consistent with our data, that I will describe a bit today, relied on a change we observed between behaviors in the correlations among neuronal firing patterns. Okay, and these sorts of changes in firing pattern correlations are evident at the single cell level here if we look at cell 73 and cell 184. These cells show very similar firing patterns during the precision pull behavior, but very different patterns during treadmill walking. So the correlations between their firing patterns are changing between the two behaviors. How could this lead to behaviorally selective pathway engagement? Well, the idea is that if the correlations among firing patterns change enough, then we can find weighted sums of those activity patterns so that during one behavior, say one weighted sum could be strongly modulated, whereas a different weighted sum here could be minimally modulated or not modulated. Whereas during a second behavior, that second weighted sum could be strongly modulated, but the first one could be weakly modulated. And if we imagine then that we have some downstream neurons that are listening to the output of our population here, then if these neurons are listening to one of these weighted sums via the intervening synaptic weights here, then these neurons could be strongly modulated during one of the two behaviors, but much more weakly modulated during the other. And so this would allow downstream neurons to respond in a behaviorally selective manner, even if the upstream neurons are active during really all types of behavior. And we considered this idea based on the work of a number of other groups very recently. They've shown that this sort of structure in neuronal population activity has some explanatory power in interpreting data from several different motor system areas. And this is the structure that we see in our data. So here I'm showing you 4 different weighted sums of the firing patterns from all of the neurons recorded in one of our mice. Each column here is a different weighted sum. In red, we have their modulation during precision pull, and in black, we have their modulation during walking. These weighted sums are defined by principal components. That's not super important. What is important here is just the fact that these weighted sums are strongly modulated during one behavior, but not so modulated during the other. And we can go the other direction here, find a separate set of weighted sums that is strongly modulated during the second behavior, but again, not so modulated during the first behavior. In both cases, the 4 weighted sums here account for the vast majority of the variance in neural activity. Each set here of 4 principal components collectively accounts for over 90% of the variance in the one behavior, but a much smaller fraction of the variance collectively in the other. And because what we're really concerned with here is motor cortical output, we also check to make sure the same result holds for the putative pyramidal neurons that are recorded in cortical layer 5B, which is where the subcerebral projection neurons reside. We used a model-based approach to classify neurons based on their spike waveform width into either putative inhibitory interneurons or putative pyramidal neurons. And we found that the dramatic change in firing rate correlations held when we specifically looked at the wide spiking putative pyramidal neurons that were recorded in layer 5B. OK, so to summarize here, I'm plotting an alignment index that measures the change in activity correlations between contexts. It's basically measuring how much overlap or alignment there is between sets of top principal components between 2 contexts. For our neural data in aggregate here, the overlap between the 2 contexts is down around 10%, whereas by random chance, we would have expected it to be about 90% if the correlation structure was constant between the 2 behaviors. And we saw a similar sort of difference when we restricted ourselves to the wide spiking putative pyramidal neurons recorded in layer 5B. When we measured this overlap for muscles, though, we saw a much higher value. We saw a value around about 65% on average. So there is some change in the correlations for muscle activation patterns between the 2 behaviors. It's not as big as the change we saw for the neural activity patterns. So this could imply that the changes that we see in the neural firing pattern correlations aren't just a simple consequence of the fact that the muscles are doing something slightly different between the 2 behaviors. So we think that this provides a sort of a reasonable working hypothesis for how the differential effects of fast time scale inactivation we observed could be mediated. The idea here is that the short latency pathways that drive muscle activity during the trained behavior may only be sensitive to certain patterns of motor cortical output. So it's as if motor cortex sort of operates in different control modes during different behaviors, with only some of its output during movement being involved in directly tugging on muscles. OK, so I'll switch gears here and briefly describe how this sort of approach has been clarifying in another project in the lab. Here, a graduate student, Claire Werner, has been working with me using the mouse to revisit some previous work in cats and primates, actually, about how the motor cortex enables the simultaneous co-contraction of antagonist limb muscles. Antagonist co-contraction is important to many different motor behaviors, especially when precision movements are being learned. One mechanistic idea from the literature is that cortical output, driving flexor and extensor or antagonist pairs of muscles, is just going to be summed together when the animal wants to co-contract those muscles to stabilize a joint. There's also evidence, though, that cortical output can do something special during a co-contraction. There are inhibitory cell types in the spinal cord that help ensure the alternation of antagonist muscles during joint rotation. And some really nice work, actually, from humans by Jens Bo Nielsen and others suggested that cortical output facilitates co-contraction by actually muting this inhibition within spinal circuits. So these mechanistic ideas I just mentioned, they're not necessarily opposing, but what we've been doing here is basically trying to test predictions of those ideas. We developed a behavioral paradigm in which mice perform two different tasks, a wheel-turning task in which antagonist limb muscles alternate their activation, and then an isometric task in which those same muscles have to co-contract. And here I'm showing you average activation of antagonist triceps and biceps muscles, both during the alternation task and then during the co-contraction task here. And Claire's done a lot of neural recordings with silicon probes targeted to layer 5B in this paradigm. And her analysis suggests that a unique neural strategy could be employed during co-contraction. For example, when she used linear regression to fit a weighted sum of neural activities to the alternating activation of antagonist triceps and biceps muscles, she can fit the muscle activity very well, but when she then fits the muscle activation from the other context with those same weighted sums of motor cortical activity, the fit is extremely poor. And here again, we can find weighted sums that are defined by principal components that account for the vast majority of the variance in the neural activity that that are strongly varying again in one context, but much more weakly varying in another context, just as I showed in the previous case for precision pull and walking. So this is nice, but is motor cortical output doing something specifically to spinal inhibition? That's the prediction we'd really like to test, and we're currently working on a couple of ways in order to address this. First, our newfound ability to specifically and very efficiently label corticospinal neurons with calcium indicators using retro AAVs. This is allowing us to address findings, or whether the findings from our probe recordings hold specifically when we look at corticospinal neurons themselves. So here I'm showing you cross-sections of the apical trunks of corticospinal neurons labeled with GCAM6. This imaging plane is about 350 microns below the cortical surface. And we're optically recording activity in these neurons during our two behaviors and estimating underlying firing rates using spike inference methods and so forth. And second, we're using newfound genetic access to try and get target specificity in our measurements here. So here we've partnered with another graduate student in the lab, Samaher Fajeri. Samaher has developed a transsynaptic rabies virus-based approach that allows efficient genetic access to subsets of corticospinal neurons that synapse onto particular cell types within the spinal cord. Here I'm showing you the population of corticospinal neurons that synapse onto GAD2 expressing interneurons in the spinal cord. This population includes those neurons that mediate presynaptic inhibition within spinal circuits. And coupling this with optical recording is now allowing us to see how activity within this specific projection population is modulated between alternation and co-contraction. And we're able to do so in a manner that avoids any potential perturbation to the cell physiology or the circuit function from the rabies virus infection itself. Okay, so lastly, with the last couple of minutes, I'll just talk about where I think I might be headed. And I'm particularly excited now because I think our new methods for measuring neural activity, our new genetically mediated approaches for targeting subpopulations of neurons within circuits, and our new computational tools for analyzing our data, I think these have us really well positioned to answer some meaningful questions in the near future. I think combining fast time scale and activation and EMG with opportunities for working with freely and naturally behaving rodents, this should allow us to clarify things about the real-time operation of motor circuits. And the increasing ease of large-scale electrical recordings, I think this is going to help us clarify functional relationships within those motor circuits on behaviorally relevant timescales. The development of head-fixed paradigms in which mice will perform trained forelimb movements will allow us to use optical approaches now to causally test ideas about differential functionality of weighted sums of activity or other sorts of manifold-based decompositions. And I think there are meaningful experiments that can be done here that aren't necessarily going to require us to simultaneously and with cellular resolution perturb activity in hundreds or thousands of cells, which at this point is still maybe prohibitively difficult. And then lastly, the super exciting developments from single-cell sequencing, a lot of which are coming from here at the Allen. These are really going to show us new granularity within neuronal populations. And I think coupling this with functional measurements and perturbations should really help us address, I think, in a new way, what the essential functional units within motor circuits actually are. So with that, I'll just say thanks to all the people that have helped here. Claire and Samaher are 2 graduate students in the lab that I was lucky to work with a lot. Gamal Al-Said and Jeff Seeley are formerly graduate students in Columbia's Neurotheory Center that helped with some of the analysis. Big thanks to Mark Churchland, John Cunningham, Larry Abbott, and Darcy Paterka for a lot of advice. Huge thanks to my advisor, Tom, the rest of his lab, funding sources, and thanks again to all of you for the chance to present. Really appreciate it. Thanks.
Thank you. Do we have any questions? We've got microphones.
Yeah, Andrew.
Hey.
Great talk. 2 questions. First, are there any descending corticospinal neurons that are inhibitory?
You looked at my drawing a little too carefully, I think. Sure, no. As far as I know, there are not. But yeah, that minus sign is going to require like an intervening interneuron if you
Okay.
Yeah.
And secondly, how do you think that training affects the kind of influence of motor cortex on EMGs that you see? So like, if you were to train a mouse to walk with some alternate gait, for example, do you think that you would see an effect or a larger effect?
Maybe at first, right? So it seems like from the literature that there are sort of 2 classes of behaviors, that there are behaviors that are always going to require motor cortical involvement and other behaviors that may only require motor cortical involvement sort of while they're new or being learned, right? So if you were training a mouse to do some weird kind of walking, it might have to sort of use its motor cortex to figure out how, but whether or not it needs the motor cortex after it's done that a million times is another question.
So, but in your precision pulling task, why are those animals just not overtrained? Why is it that you see an effect there?
Yeah, so, well, I mean, they're trained a lot. That behavior was, so we do those perturbation experiments after about 1,000 or 3,000 trials. The behavior is designed to mimic those that still require motor cortex, even at, 30,000 or 100,000 trials in some of these experiments that Ben Sylvetsky has done. So we expect, based on his results, that, if we looked even that far out, we'd still see an effect. That said, though, I think it's important to keep in mind that a big part, potentially extremely big part of what the motor cortex might do is drive movements that are in some sort of ephemeral, pre-automatic sort of state before they become super stereotyped, right? So, in thinking about, motor cortical dependent behavior, thinking about a regime where behaviors are still being learned, I think is somewhat appropriate.
Are there other questions?
Andrew, right here.
Hey.
Great talk. Just a quick question. Can mice do the co-contraction, the isometric pull with mucimolin activation or some other perturbation of M1?
Yeah, so there is an effect. They still kind of do it. It doesn't, the mucimol injections don't completely destroy the, they don't get 0 rewards after mucimol injection, but they do get, a much smaller number of rewards. And sort of, we've yet to sort of analyze the EMG to the extent where we can really say what they're doing to get rewards in those remaining cases, how it resembles what they do, in the sort of normal state. But there is an effect from an activation.
All right, let's thank Andrew again.
Thanks.